Hello, my name is Darshan, and I'll be giving you a mock lesson today on A-level chemistry, specifically on the topic of amino acids. Now, looking at the slide here, there are three learning objectives for this topic. The first one is to describe the acid and basic properties of amino acids and the formation of Switzer ions. The second one is to look at the formation of peptide bonds between amino acids, right here. And the third one would be to look at the results of electrophoresis on the mixture of different amino acids. And a cheeky little fun fact about why this topic is so important. Well, there are 20 different amino acids that are used by all living and 9 of these amino acids are essential, which means they can't be produced in the body, and so they must be obtained in the diet, while the rest are non-essential, which can be produced endogenously in the body. Yep, so good to know. So this is the outline of the class for today. It's a three-step approach, starting off with the theory lesson right here, followed by attempting a few past year questions, which are so important in consolidating knowledge for this particular topic. And I don't think we have time for this today, but this is what I would normally do, which would be to review challenging questions as well. So it's a three-step approach for every lesson. All right, so let's just dive right in. So when we look at amino acids, let's consider a typical structure of the amino acid. The first one would be it has a carboxylic acid group. It has an amine group. It also has an R group, which is quite variable. It can be neutral, basic, or acidic, as well as a single hydrogen atom. So these are the four components which make up a single amino acid. Alright, so let's just have a look at the structural formula of this compound here, right? And if I were to draw it, it would look something like this. You have the R group, which is a variable group. Remember, it can be anything at all. You have the amine group at the top, carboxylic acid group at the bottom, and a single hydrogen atom over here, right? And let's have a look at something called Switzer ions. And Switzer ions basically are the result of hydrogen transfer, all right? So it's the same molecule, but let's just transfer this hydrogen up here. So we are essentially moving the hydrogen from the carboxylic acid group for it to join with the amine group, and you would form a compound like this, right? So remember, hydrogen atom is positive in charge. Oops, sorry, let me just get this. Yeah, so hydrogen atoms are positively charged, and so when they join the amine group, it results in a positive ammonia group. And since the carboxylic acid group loses a positive charge, a single positive charge, it becomes negatively charged. So one becomes positive, one becomes negative. And we can see it perfectly cancels the charge out. And so that's exactly what a Switzer ion is. It's when both cations and anions are present in the same compound, right? So you have a positive charge and a negative charge, which completely cancels each other off, and that's what we call a Switzer ion. And we'll have a look at an application of a Switzer ion in some past year questions. Moving on to buffer solution. So a buffer solution is basically a solution when small amounts of acid is added. So remember, acid consists of many hydrogen ions, and so when you add hydrogen ions to a Switzer ion, for example, positive charges would always want to interact with a negative charge, and so you would form a complete carboxylic acid group. In your ammonia, your positively charged ammonia group would remain unchanged. Similarly, when small amounts of base is added, so remember, bases are in particular hydroxide ion is negatively charged, it would want to interact with the positive charge right here, and so the hydrogen ion, which is positively charged, would interact with this hydroxide ion to form water, and your negatively charged carboxylate group would remain unchanged. So it's a matter of understanding what atoms or what ions interact with one another. So remember, opposite charges attract, and likewise, same charges would repel each other. Alright, I'm just gonna stop right here and check that we're on the same page. Alright, so let's just take a step back, and I'd like you to know, I'd like you to let me know if you have any problems with what we've discussed so far. So it's really important that we're on the same page and that you get the most out of the lessons that we have here together. And if I do get the thumbs up from the students, then we're good to proceed. So in terms of the isoelectric point, it's basically the pH in which there is no overall charge on a particular amino acid in its aqueous solution. So definitions in chemistry are very, very important because it comes up frequently in past exam questions. So this is the typical structure of an amino acid right here, specifically in its Zwitter ion form. And remember, Zwitter ions are neutral because the positive charge would cancel out the negative charge, like I mentioned before. Alright, let's just skip peptide bond formation for today and jump straight to electrophoresis. So it's a technique that's used in the lab to separate, purify, and identify proteins, amino acids, and DNA. Alright, and you accomplish this by using an electric field that exists between a positive and negative electrode. So this is typically done when there's with the use of a battery. You have a positive terminal on the left and a negative terminal on the right, and you have the compound sitting right in the middle. The moment you connect the circuit together, you will start to see some separation, and that separation is key in what we have to consider here. All right, so let's just have a look at some of the factors that influence the separation. The first one is charge, and the second one is size. So if we think about charge, the charge of your amino acids would influence the direction, the speed, and the distance. So in terms of direction, a positively charged amino acid would move to the negative terminal, which kind of makes sense because opposite charges would attract, right? In terms of speed, the higher the charge, the faster it moves, right? And distance as well, which is very similar to speed. The higher the charge, the further it moves. In terms of size would only affect one aspect, which is speed. The larger the amino acid, the slower it moves. Alright, so this is critical. It's a very critical piece of information that you need to use in answering some of the past year questions. Alright, in terms of factors that affect direction of travel, which is very similar to what we previously discussed. In terms of direction of travel, it's always the charge of the amino acid and the pH of the buffer that's being used, which would dictate the direction of travel. In terms of speed, it's always the magnitude of the charge. And remember, when we talk about charge, we talk about overall charge. And we'll have a look at this in a moment. Magnitude of the overall charge, molecular mass, the voltage that's being applied between the electrodes, as well as the temperature right here. Alright. And just a quick reminder on gel electrophoresis. When you have electrodes, a positive electrode is called an anode, and a negative electrode is called a cathode. Alright, so that's it for the theory. It's a lot to cover, I understand. So I'm just gonna pause right here and give you a moment to consolidate everything, try to brain what we've discussed. And before we move on, I would just like to clarify that we're on the same page. And if you have any questions, this is the moment in which I address those questions. All right. All right. So most students have they often encounter difficulty in predicting what kind in predicting the way in which they'll be tested formally in the actual exam. So this is where tutoring lessons can come in handy, because I aim to bridge the gap between what you're currently studying and what you should expect to come out in the in actual exams. So bridging that gap between theory and practicality. All right, so moving on to some past paper questions. All right, let's have a look right here. All right, so what do we have here? The diagram, let's just read the question again. The diagram shows the results of electrophoresis on a mixture of the amino acids glycine, lysine, glutamic acid at pH 7.0. The structures of the amino acid at pH 7.0 are shown as below. So we have 3 different amino acids here. All right, and the question is to identify the amino acids responsible for the spots labeled R, S, and T. All right, so let's just have a look at this. All right, so this is before you turn on the charge, or before the circuit is connected, and this is after the circuit has been connected. All right, so you can see there is separation for some of the amino acids, right? And we can see that S does not move, it stays in that same spot, whereas R and T moves to opposite sides. All right, now let's just have a look at the structural formula up here. Remember what I said about charge? It has to be, we have to take into account the overall charge for a particular molecule. So we can see that this part here has a positive 1 charge, and this part here has a negative 1 charge. And so the overall charge for glycine is 0, because positive 1 and negative 1 cancel each other out, so we're left with 0. Similarly, for lysine, the overall charge is positive 1 here. We don't have any charge all the way here, but we do have a charge at the side here. This is positive 1, and this is negative 1. So the overall charge for this one, for lysine, would be positive 1. Why is it positive 1? Because positive 1, negative 1 would cancel each other out, and so you're only left with this particular positive 1 charge. All right, let's do the last one. This is positive 1. You don't have anything over here. This is a negative 1 charge. This is a negative 1 charge, and so the overall charge would be -1. Why is it -1? Positive 1, negative 1 cancel each other out. You're left with a negative 1 charge. All right, so the ones highlighted in red are your overall charges for glycine, lysine, and glutamic acid, respectively. All right, so remember, opposite charges attract, same charges or like charges would repel each other. So we can see that glycine has a 0 charge, which means it's neutral, and so it's neither attracted to the anode nor the cathode. But before that, let's just label this pretty quickly. A positive terminal would be called an anode, a negative terminal would be called a cathode. All right, and so like we mentioned before, glycine has an overall charge of 0, so it's not attracted to the anode nor the cathode, and so S would have to be glycine. All right, let's consider lysine here. So lysine has an overall charge of +1. So if this is +1, it is attracted to the negative terminal. It is attracted to the cathode, which means this has to be lysine because it's connected to the cathode or the negative terminal. So T would be lysine. Finally, to consider glutamic acid, it has an overall charge of -1. A negative charge would be attracted to positive electron, which is also called the anode right here. And so R has to be glutamic acid. All right, and so we can see that this question is worth 3 marks, and we've gotten all 3 marks here, right here. All right, pretty good. So I'm just going to stop here, see if we're on the same page. Any questions for me so far? All right, if no questions, can you give me a thumbs up before we move on? All right, let's just move on to the next one, which is on electrophoresis. All right, so a mixture of amino acids, right, can be separated using electrophoresis, and the setup is shown in the diagram below. So you have the power supply of the battery, and electrolyte, and an amino acid which is placed right in the center right here. And on both sides, it's also soaked in buffer solution. So the question is, when the power supply is switched on, some amino acids may not move but remain stationary. Suggest an explanation for this observation. All right, so let's think about it. If your compound or your amino acid mixture is in the center and it doesn't move to either side, which means it stays stationary, there are a few reasons in which it might stay stationary. So remember the key principle: opposite charges attract, and then like charges repel. So this is a very key, it's a key concept that you have to keep in mind of, okay? So in order to move to either side, it must have a charge. If it doesn't have a charge, meaning to say if it's neutral, or if the overall charge is 0, that's when it does not move. So we can say for the first one, just having a look, we can see that this question is worth 2 marks. We need 2 points, 2 separate points. So the first one would be the amino acid is neutral, which makes sense, because if it's neutral, it's not going to move anywhere. It's not going to move towards the anode, but it's also not going to move towards the cathode. All right, the second one would be because of the splitter ion. So remember, a splitter ion is when you have a positive charge and a negative charge on the same molecule. So if they have a positive charge and a negative charge on the same molecule, it will cancel each other out to form a neutral ion. So that would be our second explanation. The amino acid is in its splitter ion form. And because it's in its splitter ion form, the charges cancel each other out. It is neutral, and so it remains stationary. And so we have 2 marks right here. All right, now moving on to question B, it's worth 1 mark. The amino acid glycine has the formula given as such. Identify a species formed on the filter paper if glycine moves to the left or towards the positive end of the filter paper. So remember, positive or the positive terminal is called an anode. And bringing back this principle over here, opposite charges attract. So if the anode is positively charged, which means the amino acid that moves towards the anode would have to be negatively charged, right? So let's consider this formula right here. We have to convert this. To form this negatively charged ion, right? And the way we do that is through hydrogen transfer. I'm just going to draw this out for you so it's a lot more visible. So we have C, we have CO2H, we have a hydrogen, we have NH2, which is an amine group, and we also have another hydrogen, so this acts as the R group right here. All right, so how do we form a negatively charged ion? We can do two things, right? Two options, I would say. The first option is to add a H plus, or we can lose a H plus. So if we add a H plus to the amine group, this would form a positively charged ammonia group. But that's not what we want. We want a negatively charged ion. But if we lose a hydrogen from your carboxylic acid, you would form CO2 minus, which is negatively charged, and this is exactly what we want. And where does the H plus go? It would go on and combine with the amine group right here. All right, so but we don't have to show this this step. We just have to show the conversion of this amino acid, the original amino acid, into one which forms our negatively charged ion. And so the answer for this one would be as such. I'm drawing this out for you. You can see the structure of the amino acid right here. So we've lost one hydrogen ion, and so it forms a negatively charged carboxylate group, and your amine group remains unchanged. And so just to double confirm that we've got the right answer, what is the overall charge for this molecule? It is -1. This is neutral, neutral, neutral. This has a negatively -1 charge, and so the overall charge for this molecule is -1. And this -1 would move towards the positively charged electrode, also called the anode, and that's when we know that we have the correct answer. All right, feeling good so far? All right. Do you have any questions for me so far? Are we good? Okay. And so if we're good, we'll just attempt this last question for today, which is also on electrophoresis. And we're really considering the size and the charge of the amino acids right here, A, B, and C. So let's just label it. So positive charge terminal would be an anode, a negatively charged terminal would be a cathode, right? So we can already deduce straight off that A and B, since it's moving towards the left, towards the anode, which is positively charged, it has to be negatively charged for A and B, because opposite charges attract. Similarly, C moves towards the right, which is negatively charged, and so C has to be positively charged. And so that's our answer right there. A is negatively charged, B is also negatively charged, and C is positively charged. Done. Moving on to the other part, which is relative size. So remember what I said before. If your molecular mass increases, distance decreases. It's also there's also an analogy which may help you think about this. If something is pretty heavy, it's not going to move very far. So we can see that A moves the furthest. The starting point is over here, and this particular line here. A moves the furthest, and so A has to be the smallest, because something very small can move so far. If we compare B and C, we can see that C moves a greater distance compared to B, right? Yeah. So we can see that B moves the least, and so B has to be the largest size. Something that's very big, something that's very heavy, it's not going to move very far, and C would be in the in the middle. So you can either say medium or intermediate, and both these terms would be accepted right here. Alright, so we've tackled 2 different past paper questions on amino acids and gel electrophoresis. So after learning all of that, is there anything that you're still unsure of? Was there anything that you'd like me to cover or to touch on before we end the lesson today? And if not, we'll continue the next lesson attempting a few challenging past year questions to really consolidate knowledge that we've learned so far. And that should be it for the lesson today. Thank you very much for joining me, and I hope you found this helpful.